Hey there, and welcome back to the Sleepless Scientist. Tonight, we're slipping quietly into a world often overlooked, a realm where life thrives with astonishing ingenuity. We're going to explore the hidden intelligence of plants, the silent wonders that challenge everything we thought we knew about the botanical world. Before we journey deeper, take a moment to settle in. If you enjoy these quiet explorations, please consider liking this video and subscribing to the channel. It helps us continue these nocturnal scientific adventures. Now, let's begin our first chapter by stepping back in time to a peculiar discovery. Imagine the late 18th century. European naturalists, driven by an insatiable curiosity, ventured into uncharted territories. They meticulously documented every new species, every strange adaptation. But some discoveries pushed the boundaries of belief, hinting at a botanical world far more active than anyone imagined. One such discovery involved plants that seemed to devour insects. The very idea was met with skepticism. Plants were, by definition, passive. They drew sustenance from the soil and sun. How could a rooted organism possibly hunt? Yet the evidence mounted. Reports emerged from distant bogs and wetlands, places where the soil itself offered little nourishment. Here, life found a way, not through passive absorption, but through active, ingenious predation. This was the dawn of understanding carnivorous plants. The secret lay in their habitat. Bogs, fens, and other acidic wetlands are notoriously poor in essential nutrients, especially nitrogen and phosphorus. Most plants struggle to survive in such barren conditions, but for a select few, this scarcity became an evolutionary forge. Over millennia, these plants evolved a radical solution: supplement their diet by capturing and digesting small animals, primarily insects. It was a desperate adaptation, a brilliant strategy to thrive where others withered. The lines between plant and animal began to blur. Perhaps the most iconic of these silent hunters is the Venus flytrap Dionea muscipula. Its name itself evokes a sense of myth and danger, but its mechanism is pure, elegant biology, a marvel of natural engineering. The flytrap's jaws are actually modified leaves hinged along the midrib. The inner surfaces are lined with tiny, stiff hairs known as trigger hairs. These are the plant's sensory organs, its alarm system. An insect landing on a single trigger hair usually causes no reaction. But if it touches two different hairs within about 20 seconds or the same hair twice, the trap snaps shut with astonishing speed. This ensures the prey is active and worth the energy expenditure. The rapid closure isn't due to muscle, of course. It's a complex hydraulic process. Cells on the outer surface of the trap quickly swell with water, causing the lobes to flip inwards, much like a spring-loaded mechanism, trapping the unsuspecting insect. Once sealed, the trap forms a digestive stomach. Glands on the inner surface secrete enzymes and acids that break down the insect's soft tissues. This can take several days, slowly extracting vital nutrients. After digestion, the trap reopens, ready for its next meal. Only the indigestible exoskeleton remains, often blown away by the wind. It's an efficient, self-cleaning system honed by evolution. Moving from snapping jaws, let's consider the elegant, shimmering beauty of the sundews, Drosera. These plants employ a different, yet equally effective, strategy: a sticky embrace. The sundew leaves are covered in numerous tentacles, each tipped with a droplet of sticky mucilage. This dew glitters in the sunlight, attracting insects who mistake it for nectar or water. It's a beautiful, yet deadly, lure. Once an insect touches the mucilage, it's immediately stuck. The more it struggles, the more entangled it becomes. The sticky fluid not only traps but also begins to suffocate the prey. Unlike the flytrap's rapid snap, sundews react more slowly but just as effectively. The tentacles surrounding the trapped insect gradually bend inwards, further securing the prey and maximizing contact with digestive glands. The mucilage itself contains digestive enzymes. As the tentacles fully embrace the insect, more enzymes are released, breaking down the prey into absorbable nutrients. It's a gentle, yet relentless process. Now, let's journey to the world of pitcher plants. These include the elegant nepenthes of the Old World tropics and the striking Sarracenia of North America. Both use a pitfall strategy, luring insects into a deadly, liquid-filled well. Pitcher plants are masters of deception. Their modified leaves form deep, often brightly colored pitchers. Nectar glands along the rim and lid attract insects, offering a tempting, sweet reward. Below the nectar glands, the inner surface of the pitcher becomes incredibly smooth and waxy, often covered in downward-pointing hairs. This combination makes it impossible for an insect to maintain its grip. At the bottom of the pitcher lies a pool of digestive fluid. This fluid contains water, wetting agents to drown the prey, and a cocktail of enzymes and acids, much like an animal's stomach. Some pitcher plants have even evolved symbiotic relationships. Certain mosquito larvae or small spiders can live within the pitcher fluid, feeding on trapped insects without being digested themselves, a fascinating example of coevolution. Beyond the well-known, there are lesser-known wonders like the Australian pitcher plant, Cephalotus follicularis. It's a small, intricate plant with miniature pitchers, a testament to convergent evolution. And then there are the butterworts, Pinguicula. Their leaves are covered in tiny, glandular hairs that secrete a sticky mucilage, similar to sundews, but without the active tentacle movement. They simply act as passive flypaper. Perhaps the most active and unusual are the bladderworts, Utricularia. Many are aquatic, with tiny, pea-sized bladders that function as sophisticated suction traps, capable of capturing microscopic prey in milliseconds. The bladderwort creates a vacuum by pumping water out of its bladder. When a tiny organism brushes against trigger hairs at the entrance, a trapdoor opens, and the prey is rapidly sucked in by the sudden influx of water. It's the fastest trap in the plant kingdom. These carnivorous plants force us to reconsider our fundamental definitions. They are not merely passive organisms rooted in place. They are active hunters, equipped with sensory systems, rapid movements, and sophisticated digestive chemistry. The enzymes and acids they produce are remarkably similar to those found in animal digestive systems. This biochemical prowess highlights the incredible adaptability of life and the diverse paths evolution can take. Their existence in nutrient-poor environments is a testament to their ecological specialization. They fill a niche, turning a disadvantage into a unique survival strategy, becoming apex predators in their microecosystems. From the initial disbelief of early botanists to our current detailed understanding, the study of carnivorous plants has profoundly influenced our perception of the plant kingdom. They shattered the notion of plants as static and unchanging. Each species, with its unique trap and hunting strategy, is a miniature marvel. They invite us to look closer, to appreciate the intricate solutions nature devises when faced with environmental challenges. The study of these silent hunters pushes us to rethink what intelligence means in the natural world. While not conscious in the way animals are, their sophisticated responses to stimuli are undeniable. Beyond the scientific intrigue, there's an undeniable beauty in their adaptations. The glistening sundews, the elegant pitchers, the poised flight traps, each a work of evolutionary art designed for survival. Understanding these plants required immense patience. Years of observation, countless experiments, and a willingness to challenge established beliefs. Science often unfolds in these quiet, persistent ways. The silent hunters are just one chapter in the vast, untold story of plant intelligence. They are a powerful reminder that the botanical world holds countless secrets, waiting to be gently unveiled. Tonight, we've peered into a world where plants defy expectations, where they actively seek out what they need. It encourages us to look beyond the obvious, to question our assumptions about life itself. This exploration of carnivorous plants is just the beginning. In the chapters to come, we will delve into even more subtle, yet equally profound, aspects of the botanical world. Consider the sheer ingenuity, the quiet determination of these plants. They remind us that intelligence takes many forms and life in all its manifestations is endlessly resourceful. What other wonders lie hidden just beneath the surface? As we drift towards sleep, let the image of these silent hunters linger. They are a testament to nature's boundless creativity and a prelude to the ancient networks, whispering roots, and counting flowers we'll uncover next time. Until then, rest well. Our journey into the botanical world continues, moving from the visible hunters to a hidden realm beneath our feet. After witnessing the active lives of carnivorous plants, we now turn to a silent, ancient mystery. For centuries, forests were seen as collections of individual trees, each competing for sunlight and soil. But what if this view was incomplete? What if a deeper, unseen collaboration was at play? The idea began to emerge from patient observation, a vast, subterranean network connecting plants in ways previously unimaginable. Scientists started calling it the Wood Wide Web. Early ecologists noticed patterns of growth and decline that defied simple explanations. Some trees thrived even in poor soil, while others struggled despite ample light. Something else was at work. The key to this mystery lay not in the trees themselves, but in the soil, specifically in the intricate threads of fungi that weave through the earth, forming a vast, living tapestry. These weren't just any fungi, they were mycorrhizal fungi forming a symbiotic relationship with plant roots. Myco meaning fungus, rhiza meaning root, a partnership millions of years old. This partnership dates back to the very first plants colonizing land some 470 million years ago. It was a crucial step, enabling plants to access nutrients from barren soils. The fungi act as an extension of the plant's root system, vastly increasing its surface area. They are far more efficient at absorbing water and essential nutrients like phosphorus and nitrogen. In return, the plants, through photosynthesis, provide the fungi with sugars, carbohydrates, the energy currency of life. It's a classic exchange, beneficial to both partners. Beneath a single forest, these fungal networks can span miles. A teaspoon of forest soil can contain miles of fungal threads connecting countless plants. One pioneering researcher, Suzanne Simard, dared to challenge the prevailing view of fierce competition. Her work began to reveal the true extent of forest cooperation. Her experiments in the Canadian forests involved tagging trees with radioactive isotopes. This allowed her to trace the movement of carbon, a remarkable scientific detective story. What she found was astonishing. Carbon fixed by one tree was not staying put. It was traveling through the fungal network to neighboring trees, even those of different species. Simard identified mother trees, older, larger trees, as central hubs in these networks. They were not just surviving, they were actively nurturing the forest. Through the fungal web, mother trees would share carbon with struggling seedlings, especially those in deep shade. It was a lifeline, increasing their chances of survival. This wasn't limited to parent-offspring relationships. Different species like Douglas fir and birch were exchanging resources, forming a truly integrated community. Beyond nutrients, the network carried information. When one tree was attacked by insects, it sent chemical warning signals through the fungi to its neighbors. Receiving these signals, neighboring trees would then ramp up their defensive chemistry, preparing for a potential infestation before it even reached them. A silent alarm system. This communication involves complex organic compounds, hormones, and enzymes. The fungi act as both conduits and translators, facilitating this intricate chemical dialogue. These discoveries fundamentally challenged the long-held view that competition was the sole driving force in ecosystems. Cooperation and altruism were equally vital principles. Such interconnectedness provides remarkable resilience. A forest with a robust wood wide web is better equipped to withstand disturbances like drought or disease. Not all fungal partnerships are the same. Ectomycorrhizae, often forming sheaths around roots, are common in temperate forests, linking many tree species. Endomycorrhizae, on the other hand, penetrate plant cells directly. They are found in a vast majority of plant species, including many agricultural crops and grasses. This diversity highlights the ancient coevolutionary dance between plants and fungi, adapting to different environments and nutrient requirements over geological time. Uncovering these hidden worlds required innovative methods. Isotope tracing, as Simard demonstrated, was crucial for visualizing the flow of resources. Genetic sequencing further revolutionized the field. Researchers could now identify the specific fungal species involved and map their connections with unprecedented accuracy. By combining these techniques, scientists began to create intricate maps of the wood wide web, revealing its astonishing complexity and reach. The evolution of these scientific tools allowed us to peer into a realm once invisible, transforming our understanding of forest dynamics from a reductionist view to a holistic one. This knowledge carries profound implications for conservation. Clear cutting, for instance, severs these vital connections, impacting the entire ecosystem's health and recovery. Understanding the wood wide web advocates for practices that preserve the integrity of these underground systems, like selective logging or leaving legacy trees. The forest is not just a collection of trees. It functions more like a superorganism, a vast, interconnected entity where cooperation is as fundamental as competition. This biological phenomenon offers a profound metaphor for community and interdependence, inviting reflection on our own relationships and the hidden links that bind us. By revealing the intricate, ancient dialogues occurring beneath our feet, we cultivate a deeper respect for the subtle power and wisdom embedded within natural systems. These discoveries have reshaped our understanding of forest dynamics, moving away from individualistic perspectives to one that appreciates holistic, networked nature. Plants emerge not just as passive elements, but as central actors in a vast, living tapestry of communication and exchange, essential for all life on Earth. This journey of discovery, from initial skepticism to groundbreaking revelations, highlights the quiet persistence of science. It's a slow, deliberate unveiling. There is immense wonder in recognizing these unseen worlds, these silent conversations happening all around us, just beneath the surface. It's a shift in perspective from viewing nature as a battleground to appreciating it as a complex, cooperative dance orchestrated over millennia. These hidden dialogues beneath the soil are just one facet of the plant world's astonishing capabilities. There are still so many layers to peel back, so many stories to hear. Next, we will explore how plants, despite their stillness, possess a remarkable sense of time, counting the days and remembering the seasons, a silent, internal clock guiding their lives. We explored the fungal networks, a vast, ancient communication system, but beneath the soil, another, even more direct conversation unfolds, often overlooked. This is the secret language of whispering roots, a chemical dialogue happening plant to plant, independent of fungal intermediaries. For a long time, scientists suspected plants could sense their neighbors. Unexplained growth patterns, competition, or cooperation hinted at something more. How do you listen to a conversation happening underground in total darkness through a dense medium like soil? It required ingenuity. The answer lies in root exudates. These are chemical compounds released by roots into the surrounding soil, a plant's way of speaking. This chemical vocabulary is incredibly diverse. It includes sugars, amino acids, enzymes, and specialized signaling molecules. Scientists began the painstaking work of identifying these specific signaling molecules, each carrying a different message. Take strigolactones, initially known for promoting fungal growth, they also act as direct signals between plant roots. Phenolic compounds, another class of exudates, can influence nutrient availability and even inhibit the growth of competing plants. Phytohormones, traditionally known for regulating internal plant processes, are also released by roots to communicate externally. But how do roots listen? Plants possess specialized receptors on their root surfaces, finely tuned to detect these chemical whispers. These receptors allow a plant to perceive the presence of nearby neighbors, even before their leaves cast a shadow. Is this neighbor a competitor for light and water? The root exudates provide crucial information, allowing the plant to adapt its growth. One powerful form of communication is allelopathy. Some plants release toxins into the soil to suppress the growth of rivals. The black walnut tree is a classic example. It produces juglone, a chemical that can inhibit the growth of many other plant species. This isn't malice, but a highly effective evolutionary strategy, ensuring the black walnut's dominance in its local environment. But root communication isn't always about competition. Roots also engage in cooperative dialogues, sharing information and resources. Some plants signal to share nutrients in times of scarcity or even warn neighbors of impending stress like drought or disease. Early experiments with pea plants demonstrated this. When one pea plant was stressed, it could warn its neighbors through its roots. Upon receiving these signals, a plant's root system might alter its growth patterns, extending towards nutrient-rich areas or away from competitors. It could also upregulate its nutrient uptake mechanisms, preparing for a period of scarcity based on its neighbor's warning. Furthermore, plants can prime their defense responses. A root signal might trigger the production of defensive chemicals in a neighboring plant. This direct communication also heavily involves the rhizosphere, the tiny zone of soil directly influenced by root exudates. Within this zone, root signals shape the microbial community, attracting beneficial microbes and deterring harmful ones. Unraveling these complex interactions required sophisticated tools, moving beyond simple observation to precise chemical analysis. Techniques like GCMS allowed scientists to identify volatile organic compounds released by roots, even in minute quantities. For non-volatile compounds, liquid chromatography provided similar insights, separating and identifying the heavier molecules. Molecular biology, too, played a crucial role, identifying the genes responsible for producing these signals and their corresponding receptors. This fusion of chemistry and biology transformed our understanding, shifting root communication from a poetic concept to a tangible biological reality. The picture that emerged was of a dynamic, constantly active underground world, a hub of intelligent decision-making. This research challenged anthropocentric views, revealing that plants are not passive, but constantly processing information and responding. The implications for sustainable agriculture are profound. Understanding these root dialogues can enhance crop yields naturally. By leveraging plants' innate communication, we might reduce reliance on synthetic fertilizers and pesticides, promoting healthier soil. In ecosystem restoration, this knowledge is vital. Reestablishing healthy root communication can accelerate recovery in degraded lands. This deep dive into root whispers expands our appreciation for the sophistication of plant sensory perception. It encourages a more humble observation of nature's subtle brilliance, a world of constant, silent activity beneath our feet. Roots are not merely anchors or conduits for water. They are active agents mediating a vast, silent conversation. This conversation underpins the health and resilience of entire ecosystems, a hidden foundation for all terrestrial life. The world beneath the soil, once thought static, pulses with life and communication, a complex intelligence still revealing its layers. As the whispers fade, we prepare to witness another marvel: how plants, despite their stillness, possess an astonishing sense of time, counting the days and remembering the seasons. As the whispers of roots fade, we turn our attention to another profound aspect of plant life: their astonishing sense of time. How do plants, rooted and seemingly still, know precisely when to bloom, when to set fruit, or when to prepare for winter? This question has intrigued observers for centuries. Early naturalists noted that certain flowers opened and closed with remarkable regularity, almost as if following an invisible clock. They were seeing a fundamental biological rhythm. Carl Linnaeus, the father of modern taxonomy, even attempted to create a flower clock in the 18th century. By planting species that bloomed at different hours, he imagined a living timepiece marking the passage of the day. But a flower clock only reflected a deeper, internal mechanism. The real scientific mystery was not just that plants kept time, but how they did it. What was the sensory input, and what was the biological machinery? The answer, scientists eventually discovered, lay largely in the subtle shifts of light and darkness throughout the day and night. This phenomenon became known as photoperiodism, the plant's response to relative day and night lengths. Early experiments in the 20th century were surprisingly simple yet profound. Researchers exposed plants to varying periods of light and darkness, then meticulously recorded their flowering responses. A crucial breakthrough came with the discovery that it wasn't the length of the day that mattered most for many plants, but rather the unbroken period of darkness. The night, not the day, was being measured. Some plants, like chrysanthemums, are short-day plants, requiring a long, uninterrupted night to flower. Others, like spinach, are long-day plants, needing short nights. This precision was astonishing. Even a brief flash of light during the critical dark period could prevent a short-day plant from flowering or induce a long-day plant to bloom prematurely. The plant's internal clock was incredibly sensitive. This led to a new question: what cellular machinery allowed plants to detect and respond to these subtle differences in light and dark? The search began for the photoreceptors responsible. In the mid-20th century, scientists identified a remarkable family of proteins called phytochromes. These molecules were the plant's eyes, specifically designed to detect red and far-red light. Phytochromes exist in two interconvertible forms: PR, red-absorbing, and PFR, far-red absorbing. Red light converts PR to PFR, while far-red light converts PFR back to PR, or PFR slowly reverts to PR in darkness. The ratio of these two forms acts as a biological switch, signaling to the plant the length of the day and, crucially, the duration of the night. This sophisticated system allows plants to read the ambient light conditions. Beyond external light cues, plants also possess an internal circadian rhythm, a body clock that anticipates the daily cycle. This internal clock works in concert with phytochrome signals, creating a robust timekeeping system. The interplay between the external light signals and the internal clock ensures that plants not only perceive the current conditions, but also integrate them into a predictive model of the seasons. But how does this light information, often perceived in the leaves, translate into the momentous decision to flower in the distant shoot apex? The answer lay in a mysterious mobile signal, dubbed florigen. For decades, florigen was a theoretical concept, its existence inferred from grafting experiments. If a flowering plant was grafted onto a non-flowering one, the non-flowering plant would often begin to bloom. This suggested a chemical signal was traveling from the flowering leaves to the meristem, the plant's growth tip, instructing it to switch from vegetative growth to reproductive growth. Finally, in the early 21st century, florigen was identified. It turned out to be a small protein, a transcription factor that travels through the plant's vascular system, directly activating flowering genes. But light isn't the only seasonal cue. Some plants rely on a different kind of memory, the memory of cold. This process, known as vernalization, ensures they only flower after winter has truly passed. Winter wheat, for example, needs a prolonged period of cold temperatures to flower. If planted in spring, it simply won't produce grain that year. It needs to remember the chill. This fascinating requirement ensures that plants don't waste energy flowering in late autumn, only to have their delicate blooms destroyed by an early frost. It's a survival strategy, finely tuned by evolution. Experiments involving controlled cold exposure revealed that the duration and intensity of the cold were critical. Plants were not just sensing cold, they were accumulating a cold dose over time. The memory of this cold could persist for weeks, even months, after the temperature returned to warmer levels. This wasn't a fleeting response, it was a sustained internal record of past conditions. The molecular basis of vernalization proved to be just as intricate as photoperiodism. It involves epigenetic modifications, changes that don't alter the DNA sequence itself but affect how genes are expressed. A key player is the FLC gene, or flowering locus C. In the absence of cold, FLC acts as a repressor, preventing flowering. During prolonged cold, the FLC gene is epigenetically silenced. This silencing is stable. It persists even when temperatures rise, allowing the plant to remember that it has experienced winter. Only then can other flowering genes be activated. This molecular memory is passed down through cell divisions, ensuring that the entire plant, as it grows, retains the information about the cold exposure. It's a remarkable form of cellular memory. Plants integrate these various signals, light, temperature, and even moisture levels, to make complex developmental decisions. They are constantly processing a symphony of environmental data. The ability to precisely time flowering provides immense evolutionary advantages. It ensures that plants bloom when pollinators are active and when conditions are optimal for seed development. This precise timing prevents reproductive failure due to unseasonable frosts or dry spells. It's a testament to natural selection, refining these internal calendars over millions of years. Modern genetic and molecular research continues to unravel the intricate biochemical pathways controlling these phenomena. Each new discovery reveals another layer of botanical sophistication. From historical observations of Linnaeus's flower clock to the identification of florigen and FLC, the journey of understanding plant time has been a slow, persistent unveiling. This research challenges our traditional definitions of memory and temporal awareness. Plants, without a brain, exhibit sophisticated information processing and adaptive responses. Their inner clockwork is a profound connection to the rhythms of the earth, a silent, continuous dialogue with the changing seasons. Understanding these botanical calendars is not just academic. It has crucial implications for agriculture, helping us breed crops that are resilient to changing climates. By appreciating how plants count and remember, we gain insight into the nuanced intelligence of the botanical world, where time is a vital element in a plant's strategy for life. It invites us to appreciate the subtle yet powerful ways plants navigate their world, finding diverse and elegant solutions to fundamental biological challenges. The quiet persistence of scientific inquiry continues to reveal these wonders, blurring the lines between what we perceive as simple and complex life forms. As these silent calendars mark the passage of seasons, we are left to ponder the depth of botanical intelligence. Next, we will delve into the astonishing intelligence of the botanical world itself, an exploration that promises to challenge our very perception of consciousness. Until then, rest well. We have journeyed through the active traps of carnivorous plants, navigated the ancient fungal networks, listened to the chemical whispers of roots, and marveled at flowers that count the seasons. Each discovery peeled back a layer, revealing a botanical world far more dynamic than once imagined. These complex behaviors, hunting, sharing, communicating, remembering, begged a profound question: do plants possess a form of intelligence? The very idea challenges our traditional, animal-centric definitions of cognition. For many years, the concept of plant intelligence was met with skepticism. How could organisms without brains, without a central nervous system, process information, make decisions, or learn? Yet, the accumulating evidence from diverse fields of plant science began to paint a different picture. Plants, it seemed, were not just reacting, they were actively engaging with their environment in sophisticated ways. The debate shifted from if to how. If intelligence is defined by the ability to acquire and apply knowledge and skills, then plants exhibit behaviors that fit this description, albeit in a uniquely botanical fashion. Consider problem solving. A plant seed germinating in darkness will send its shoot upwards, seeking light, and its roots downwards, seeking water and nutrients. This seems simple, but what happens when obstacles arise? Roots, for instance, can navigate complex underground mazes, sensing gradients of moisture and nutrients, and actively growing towards beneficial areas while avoiding toxins or physical barriers. They solve the problem of resource acquisition. Plants also make strategic decisions about resource allocation. When faced with limited nutrients, a plant might prioritize root growth over shoot growth, or invest in defense chemicals rather than rapid expansion. These choices are not random. They are influenced by a plant's internal state, its genetic predispositions, and the information it gathers from its environment through a myriad of sensory inputs. It's a form of distributed decision-making where information is processed at the cellular level, leading to an integrated response across the entire organism without a central command center. Plants even exhibit forms of learning. The Mimosa pudica, or touch-me-not plant, famously folds its leaves when touched. If touched repeatedly without harm, it eventually stops responding, a process known as habituation. This isn't a simple reflex. It's a memory of previous stimuli, allowing the plant to conserve energy by distinguishing between a real threat and a harmless disturbance, such as a passing breeze. Similarly, plants can adapt their growth patterns based on past experiences. A plant exposed to a specific stressor, like drought, might remember it and adjust its physiology to be more resilient to future droughts. This memory involves epigenetic changes, where genes are turned on or off without altering the underlying DNA sequence, providing a flexible way to store and retrieve information about past environmental conditions. These sophisticated adaptive responses challenge our narrow definitions of what constitutes learning and memory, pushing us to consider the diverse ways life processes information. The intelligence of plants extends to their intricate communication with other species, most notably pollinators. Flowers are not just beautiful, they are masterpieces of interspecies negotiation. They use a secret language of color, scent, and shape to attract specific pollinators, guiding them to nectar rewards and ensuring efficient pollen transfer. Each blossom is a carefully crafted advertisement. Some orchids mimic the appearance and scent of female insects, tricking males into attempting to mate, thereby spreading pollen. This deceptive brilliance demonstrates a profound understanding of pollinator behavior. The timing of nectar production, the specific volatile compounds released, the patterns on petals visible only in ultraviolet light, all are tailored to maximize reproductive success. This interspecies intelligence is a testament to millions of years of coevolution, a silent, intricate dance between plants and the animals that serve them. Plants also employ sophisticated defense mechanisms against herbivores and pathogens. When attacked, they don't simply endure, they fight back, often with remarkable precision. They can produce a vast arsenal of chemical compounds, bitter tannins, toxic alkaloids, or digestive enzyme inhibitors, making themselves unpalatable or even poisonous to attackers. Some plants release volatile organic compounds that act as an alarm call, warning neighboring plants of an impending threat, prompting them to ramp up their own defenses. Even more astonishingly, these volatile signals can recruit the natural enemies of their attackers. For example, corn plants under caterpillar attack release compounds that attract parasitic wasps. The wasps then lay their eggs in the caterpillars, effectively turning the plant's enemies against themselves. This is a multi-layered defense strategy orchestrated by a brainless organism. The molecular and cellular basis for these behaviors is being steadily uncovered. Electrical signals, similar to nerve impulses, have been observed in plants, though they travel at different speeds. Hormone cascades like those involving auxins and gibberellins orchestrate growth and development, while others, like jasminates, mediate defense responses. Gene expression changes, often triggered by environmental cues, allow plants to reprogram their cellular machinery, adapting their physiology to new conditions. These intricate molecular dialogues demonstrate how information is processed and integrated throughout the plant, leading to complex, coordinated responses without a centralized nervous system. This challenges us to rethink the very nature of intelligence, moving beyond the animal-centric models that have long dominated our understanding. Pioneering scientists in the field of plant neurobiology advocate for a broader definition, one that encompasses the myriad ways life processes information, adapts, and thrives. They argue that intelligence is not solely about having a brain, but about the capacity for flexible, adaptive behavior in response to environmental challenges. By showcasing the astonishing ways plants perceive, react, and thrive, we unlock a deeper appreciation for their ingenuity and resilience. This exploration compels us to reevaluate our position in the web of life, recognizing plants not as passive background elements but as active, intelligent participants shaping their world. It prompts a shift in perspective towards a more profound respect for the inherent complexity and agency within all forms of life on Earth. Scientific investigation, through careful observation and experimentation, continues to reveal new layers of plant sophistication. Each discovery constantly pushes the boundaries of what we thought was possible for organisms without brains, thereby enriching our understanding of life's diverse expressions of intelligence and adaptation. The botanical world, in its quiet persistence, offers profound lessons in survival, communication, and adaptability, lessons that echo across all living systems. The astonishing intelligence of plants challenges us to broaden our minds, to look closer, and to listen more carefully to the silent wisdom embedded in the green world around us. As we conclude this exploration of minds without brains, we are left with a renewed sense of wonder. The final chapter awaits, where we will bring together these threads, reflecting on the profound implications of plant intelligence for our understanding of life itself. Until then, rest well. We've journeyed through hidden worlds, from the silent hunters of the bogs to the whispering networks beneath ancient forests. Each step unveiled a deeper layer of botanical wonder. We've listened to the secret language of roots and marveled at flowers that count the seasons, holding time within their delicate petals. We then confronted the astonishing intelligence of plants, challenging our very definitions of cognition, learning, and memory. These discoveries reveal a botanical realm teeming with sophisticated strategies, intricate communication, and profound adaptability. Tonight, as these echoes settle, we reflect on what this green story means for us, for our place within the vast tapestry of life. The plant kingdom, we now understand, is not just a backdrop to our lives, but an active, intelligent partner in Earth's delicate balance. They are the silent architects of our planet, shaping atmospheres, building soils, and sustaining the very air we breathe. This profound interconnectedness reminds us that the Earth functions as a vast, living superorganism, with plants as its vital organs. The health of this green network, its biodiversity, is intrinsically linked to our own well-being and the future of all species. This awareness calls for a deeper understanding, moving beyond mere utility to a profound appreciation for their intrinsic value. Yet this vital green world faces unprecedented challenges. Habitat loss, climate change, and human impact threaten countless species. Every lost species, every degraded ecosystem weakens the intricate web, sending ripples through the entire planet. Our newfound knowledge of plant intelligence underscores an urgent responsibility to protect these silent, powerful beings. They are not just resources to be exploited, but complex life forms deserving of respect and careful stewardship. Protecting plant biodiversity is not just about saving individual species, it's about safeguarding the very fabric of life on Earth. The ingenuity of plants also offers remarkable blueprints for human innovation, a field known as bioinspiration. From their structural designs to their chemical defenses, plants hold secrets for sustainable technologies and solutions. Imagine buildings that self-regulate temperature like leaves, or materials that self-repair like plant tissues. The quiet resilience of the botanical world provides a profound source of inspiration for a more harmonious future. By listening to the echoes in the canopy, we learn ancient lessons crucial for navigating our modern challenges. This journey transforms our relationship with plants, moving beyond a purely utilitarian view to one of deep connection. We are invited to see ourselves not as separate from nature, but as integral parts of a grand, unfolding green story. A deeper ethical consideration for the botanical world emerges, recognizing their sentience in their own unique way. This shift in perspective cultivates a profound reverence, fostering a more conscious and reciprocal existence. Our shared human endeavor with plants is one of mutual respect, learning, and harmonious coexistence. The journey of scientific inquiry itself, from initial curiosity to groundbreaking discoveries, continually expands our awareness. It's a testament to patience, meticulous observation, and the courage to challenge established beliefs. Each experiment, each new piece of data, gently peels back another layer of nature's vast, intricate design. And yet, for all we've discovered, the plant kingdom still holds immense mysteries waiting to be unveiled. This ongoing quest teaches us humility, reminding us of the infinite wonders that lie just beyond our current understanding. Future research in plant neurobiology promises to deepen our understanding of their sensory perception and decision-making. Ecological engineering, inspired by plant strategies, could lead to more resilient and sustainable urban environments. New insights into their medicinal and environmental applications could unlock solutions for human health and planetary well-being. The potential for new discoveries from the botanical world is as vast and abundant as the forests themselves. The journey of discovery is an ongoing, shared human endeavor, fueled by curiosity and reverence. As the sun dips low, we are invited to carry this awakened perspective into our daily lives. To observe with care, to protect with purpose, and to coexist harmoniously with our green companions. By listening to the echoes in the canopy, we absorb ancient lessons crucial for a thriving future. For the more we learn about plants, the more we truly understand ourselves and the delicate balance of this living planet. And so, as the moon climbs high, let the quiet wisdom of the botanical world settle within you. Sleep well, and may your dreams be green.